Welcome friends to this new lecture of soil science and technology, and in this lecture we will be uh, trying to finish the nitrogen cycle, and then we will go and discuss about biological nitrogen fixation, which is one of the most important nitrogen transformation process. So in our last lecture we covered up to ammonia volatilizations. So let us go ahead and see what is nitrification. Now nitrification, you know that it is a conversion of ammonium nitrate into ammonium nitrogen into nitrate nitrogen, and it is basically executed in two consecutive steps. In the first step you can see it is basically oxidation of ammonium with oxygen, and it is mediated by a bacteria called Nitrosomonas, and ultimately it produces nitrite, and then it produces protons, which basically causes the acidity, and also it produces water and 275 kilojoule of energy. And in the second step, this nitrite is very short lived, and it is very toxic to the plants. So this nitrate is very short lived, and again it oxidized by the help of a bacteria called Nitrobacter, and ultimately it produces nitrate and 76 kilojoule of energy. And this nitrate is the major major species, available species of nitrogen that is being uptaken by the plant. So so this conversion of ammonium form of nitrate to nitrate form, ammonium form of nitrogen to nitrate form of nitrogen is termed as nitrification, and it is basically oxidation process where Nitrosomonas and Nitrobacter bacteria plays a very important role. So let us go ahead and see what are the different factors which affect nitrification. Obviously the substrate availability is the major issue. If there is no ammonium availability, obviously there will not be any nitrification, because ammonia is the starting point, ammonium is the starting point of nitrification process. And then obviously the temperature, lower the temperature and slower the nitrification. So we have found that the 20 to 30 degrees centigrade temperature is optimal for for nitrification process. Aeration is required because aerobic environment enhances nitrification. It is an oxidation process. And moisture content, it requires well drained soil to enhance the nitrification, and generally around 60% of the of the water filled pores are optimal for for nitrification process. And also it depends on the pH as well as the type of clay. So what is denitrification? Denitrification is just opposite of nitrification. So it is a it is a reduction process where nitrate nitrogen, through a successive steps of reduction, ultimately produces this dinitrogen gas, which ultimately escapes to the atmosphere. So if you can see here, it is basically loss of nitrogen by sequential reduction of nitrate in soil. So the starting point obviously here is the nitrate, and here you can see the valence state of nitrogen here is +5 in case of nitrate, and as a result of oxygen continuous sequential reduction, there is a continuous loss of oxygen, as you can see here. And ultimately the second step, it forms the nitrate ions, and the third step is nitric oxide gas, and then the nitrous oxide gas, which is very important greenhouse gas, and finally it produces the dinitrogen gas, and this dinitrogen or along with nitrous oxide and this nitrogen escapes into the atmosphere. So this is the this is denitrification process, and it is also mediated by different microbes, and because the microbes basically, and it basically occurs in anaerobic condition, because microbes utilize oxygen in the nitrate for respiration, thereby reducing them. So in the reducing condition, when there is no oxygen available for these microorganisms, they uses these nitrate for their oxygen, and ultimately they reduce this nitrate to dinitrogen, which ultimately escapes into the atmosphere. So, end product of denitrification depends on the bacteria involved and soil conditions obviously, and low lying organic rich and oxygen depleted zones are more prone to denitrification, and denitrification results in loss of valuable nitrogen in nutrient management. Obviously, you can see the nitrogen, the final the end product is nitrogen which is being escaped to the atmosphere. So, it is useful in removing the nitrogen from wastewater. So, this is the only plus point which this denitrification process has. So, guys, we have we have completed this denitrification process also. So, let us go ahead and see. Okay, no, we have to, there are some pictures of denitrification. You can see there is a temporarily waterlogged soil. Obviously, in the waterlogged soil, all the pore space will be filled up with water. So, there will be there is very little amount of oxygen which is present in the pores. All the oxygen, or in other words, all the air will be removed by water, and as a result of that, all the microbes, the microbes basically they are facultative anaerobes. These facultative anaerobes will basically use this nitrate as a terminal electron acceptor, and then it converts this nitrate into dinitrogen. So, also you can see here the schematic diagram of denitrification in flooded system, and obviously please remember that the deep application of fertilizer helps in loss of nitrogen in flooded system. So, this is this is basically to give you an idea about denitrification, and remember that denitrification, as the time passes, the nitrogen concentration, and if you see the time in hours, the relation between the time in hours and the nitrogen concentration, you can see here the nitrous oxide gas and this nitrogen gas forms, and obviously this is the time after the submergence. So, after keeping the soil moist, so you can see the both nitrous oxide as well as nitrogen gas, their concentration is increasing, and obviously due to the absence of oxygen, the concentration of nitrate is getting decreased. So, this is basically the denitrification process, and let us see what is the Anamox process. The Anamox process is basically anaerobic oxidation of ammonium yielding to nitrogen gas. So, it is anaerobic oxidation of ammonium. So, that is the Anamox process, and is basically caused by unusual bacteria phylum Planktomycetes, and it occurs in redox transition zones like upper layer of saturated soil or rhizosphere, etc. Because you know that I have told you in our submerged soil lecture that there is a very thin oxidized layer which is present in the saturated soil, especially in submerged soil, and these Anamox process basically occurs in that redox transition zone, because it is this zone basically lies above the reduced zone which is present below. And so, basically there are two steps, one is aerobic and another is anaerobic. So, in the first step you can see this ammonium is getting oxidized to form nitrite, and it is creating the protons and water, which is an aerobic oxidation process. In the second step, obviously these nitrite which is generated in the first step will react with ammonium to produce dinitrogen as well as water. So, if we combine both these we can see this ammonium plus oxygen. So, it is an oxygen, it is an oxidation of ammonium, and ultimately it produces nitrogen and then protons and water. So, it is again the oxidation of ammonium in anaerobic condition, and ultimately the production of dinitrogen or nitrogen gas. So, this is called the Anamox process. So, nitrogen loss and environment, obviously nitrogen loss by denitrification, and Anamox, obviously in case of Anamox also nitrogen dinitrogen forms, and which ultimately escapes to the atmosphere. So, nitrogen loss by denitrification and Anamox is detrimental to plant growth in soils, whereas it is beneficial in wastewater management. Obviously, we want to that nitrogen which is present in the wastewater getting to be removed. So nitrous oxide and nitric oxide are greenhouse gases, and and they cause acid rain and ozone depletion, etc., because this nitrous oxide, when they went to atmosphere, they also create this acid rain and all these things. So slow release nitrogenous fertilizer and deep placement of fertilizer also can reduce the loss of nitrogen, because the slow release nitrogen fertilizer, they control the release of nitrogen, because nitrogen is highly soluble in water, especially the nitrate, and this deep placement, basically it helps them to reduce the reaction with the atmosphere and ultimately control the nitrogen nitrogen mobility. So this is how we can we can reduce the loss of nitrogen. And so guys, we have finished this nitrogen cycle, and let us go to our next topic, that is biological nitrogen fixation. It is very important important topic of nitrogen, so that's why we will discuss this solely as a topic. So let us see what are the different concepts which we will cover in this lecture, in this topic, that is what is the biological nitrogen fixation and what is the importance of biological nitrogen fixation, then the mechanism of nitrogen fixation, then different fixation system. We will discuss different fixation systems, and also we will be discussing different factors which are affecting the nitrogen fixation. So what is the biological nitrogen fixation? Now it is basically, next to plant photosynthesis, biological nitrogen fixation, remember, is the most important biochemical reaction for life, because this in the biological nitrogen fixation converts the inert dinitrogen gas of the atmosphere to reactive nitrogen that becomes available to all forms of life through the nitrogen cycle. So basically it is a conversion of nitrogen gas which is present in the atmosphere to some reduced form or organic forms of nitrogen, and ultimately, so by and this process is mediated by different microorganisms. So that is why it is called biological nitrogen fixation. We are fixing the atmospheric nitrogen into the soil and into the body of different microorganisms. So this is why it is called biological nitrogen fixation, and the process again is carried out by limited number of bacteria, including several species of Rhizobium, Actinomycetes and cyanobacteria, formerly termed as the blue green algae, and we will discuss that later on. And what is the importance of biological nitrogen fixation? Well, basically due to the biological nitrogen fixation, it helps to increase the yield of the crop, and it reduces the need of manufacturing the nitrogenous fertilizer, and because when there is a formation of when there is a biological nitrogen fixation, obviously the need for adding supplemental inorganic fertilizer, nitrogenous fertilizer, is reduced, and therefore lowering the amount of reactive nitrogen circulating into the environment, and ultimately due to this there is reduction in nitrous oxide emission, which is an important or very dangerous greenhouse gas. So this is the basic importance of biological nitrogen fixation, and let us see the mechanism of nitrogen fixation. Obviously, regardless of organisms involved, the key to biological nitrogen fixation is the enzyme nitrogenase. So biological nitrogen fixation is mediated through an enzyme called nitrogenase, which catalyzes the reduction of dinitrogen gas to ammonia, and you can see here here nitrogen is getting due to the nitrogenase enzyme complex, which basically contains iron and molybdenum, and they are basically reduced to form ammonia. The ammonia in turn combine with organic acids to form the amino acids and ultimately proteins. So this ammonia, when reacts with organic acids, ultimately forms the amino acids, which in turn develop proteins, and because protein is basically polymer of different it is made of these amino acids, and the site of nitrogen reduction is the enzyme nitrogenase, a complex constituting protein complex consisting of two proteins, the smaller of which contains iron, while the larger unit contains the molybdenum, sulfur and iron. We will see that in the next slide. So, if we go to the next slide, you will see there is a nitrogenous enzyme complex. Obviously, there are two units. The first, the smaller unit is basically you can see here, and the larger unit basically contain, the smaller unit basically contain iron, whereas the larger unit is basically contains iron, molybdenum and sulfur. So, let us see how these nitrogenous enzyme complex uh, takes part in nitrogen, biological nitrogen fixation. So, you can see these two proteins or these two unit of this nitrogenous complex, the larger protein converts ammonia to the larger protein converts atmospheric nitrogen to ammonia. You can see here the atmospheric nitrogen is getting converted to ammonia here using the electrons which is provided by the smaller protein. You can see electron is moving from the smaller protein to this bigger protein, and also as a result of this conversion, hydrogen gas is produced, and energy from ATP conversion to ADP. Obviously, you can see energy from ATP conversion to ADP along with electrons from ferredoxin reduces the Fe3 in the smaller Fe protein. So, in the smaller Fe protein, you can see the Fe3 is getting reduced to Fe2, and this reduction of Fe2 is basically mediated through this conversion of ATP to ADP or adenosine triphosphate to adenosine diphosphate, and also from this conversion of ferredoxin reduced to reduced version of ferredoxin to oxidized version of ferredoxin, and ultimately iron 3, that is ferric, is getting reduced to ferrous, and ultimately there is a movement of of this electron to the larger protein which contains the molybdenum, iron and sulfur. So, the molybdenum, iron, sulfur cluster in the large protein capture nitrogen from the air, while the iron in the cluster receives the electrons provided by the small protein, so that nitrogen can be reduced. For the reduction of nitrogen, you require electrons, and this electron is basically moving from the smaller protein to this larger protein, and ultimately it is reducing the nitrogen to ammonia. And this nitrogen fixation in nature may be limited by the insufficient supply of sulfur, iron, molybdenum and phosphorus. It is quite natural because this nitrogenous complex is made of all these compounds, and also phosphorus is an important element of ATP. So, any insufficiency or deficiency of any of these element will cause the nitrogen fixation process to to stop. So, these are some important factors for nitrogen important factors for nitrogen fixation, and I have told you the mechanism of nitrogen fixation. So, let us go ahead and see what are the other important and exciting facts about nitrogen fixation. Obviously, you remember that the dinitrogen has to be converted to ammonium. So, this triple bond between this nitrogen has to be broken, and this this requires a great deal of energy, and obviously the microbial process is greatly enhanced when it is carried out in association with the plants which can supply energy from the photosynthesis. So, nitrogenous obviously is getting destroyed by free oxygen, and so the organism that fix nitrogen must protect the enzyme from exposure to oxygen, and the fixation takes place in the root nodules which has leg hemoglobin, and this leg hemoglobin basically binds the oxygen to protect the nitrogenous. So, if the oxygen is if the oxygen comes in contact with nitrogenous, it will get destroyed and this biological nitrogen fixation will not occur. So, this leg hemoglobin basically binds oxygen to prevent it to oxidize in nitrogenous, and the reduction reaction is end product inhibited. Obviously, an accumulation of ammonia which is the end product will inhibit nitrogen fixation. Also, too much nitrate in the soil will inhibit the formation of nodules, because when there is an abundant amount of nitrate which is already present in the soil, so there will not be further nitrogen fixation, and obviously the accumulation of ammonia will also reduce the rate of production for the end products. So, nitrogen fixing organisms have a relatively high requirement for molybdenum, iron, phosphorus and sulfur. This is quite obvious, and let us see what are the three different fixation systems. The three different fixation systems are symbiotic fixation with legumes, then symbiotic fixation with non-legumes, and thirdly it is non-symbiotic fixation. So, what is a symbiotic fixation with legumes? Plants of legume family, that is Leguminosae family, are famous for their distinctive ability to provide the major biological sources or fixed nitrogen agricultural soil. So, they are basically known for their biological nitrogen fixation ability, and they basically do that because their association with several genera of bacteria, you know, in the subclass of alpha proteobacteria, collectively termed as rhizobial bacteria, and these bacteria include species in the genera basically Rhizobium, then Mesorhizobium, Bradyrhizobium and Encypher. So, the legume plants and rhizobia basically forms a relationship, that is symbiotic relationship. It is a mutual benefit relationship in which the host plant supplies the bacteria with carbohydrate for energy, and the bacteria reciprocate by supplying the plant with reactive nitrogen with which to make the essential plant compounds such as protein and chlorophyll. So, basically this is a symbiotic relationship. The plant supplies the bacteria with carbohydrates for their energy, which is required to for breaking the dinitrogen, and also these bacteria will give them the plant this reactive nitrogen, which is required for their production of their proteins and as well as chlorophylls. So, in the conversion involving many specific signaling, this conversion is a it is a complex procedure and it involves many specific signaling compounds, and the rhizobial bacteria find and infect the legume plant root hairs and colonize in the cortical cells and form root nodules. So, these bacteria basically forms root nodules in the roots of the leguminous plant, and that serve as the seat of nitrogen fixation. So, these root nodules are basically the seat for nitrogen fixation, remember that. So, you can see first in the first picture you can see root nodules of sun hemp or dhencha used as a cover crop or for adding nitrogen into the soil. Root nodules of sun hemp as a cover crop to add nitrogen in soil, and the second photograph you can see cross section of nodule in a soybean plant, and it basically shows the red color of the oxygenated leg hemoglobin, and finally you can see in the third picture you can see the single plant cell within soybean nodules, which shows that it is stuffed with the Bradyrhizobium japonicum bacteria, and remember that Bradyrhizobium japonicum is responsible fixing nitrogen in association with the soybean plant, and you can see these are the numerous Bradyrhizobium japonicum bacteria. So, you can see some example, this again the pea plant, and you can see the root nodules and these Rhizobium leguminosarum nodules. So, pink color in the leg hemoglobin, a protein that carries pink color basically suggests the presence of leg hemoglobin, which is a protein that carries oxygen to the bacterioids, and again this is again the soybean root nodules. So, these root nodules are the basically the sites where different this nitrogen fixation basically takes place. So, remember that specific microorganism can be associated with specific plant for fixing these atmospheric nitrogen. That means, this rhizobium and legume symbiotic relationship is very specific. A specific organism can form symbiotic relationship with a specific species. So, you can see here, just to give you some example, Rhizobium lupini, it can produce the root nodules in lupinus or lupins, and Azorhizobium species, they can produce stem nodules of Sesbania rostrata. This Sesbania rostrata is actually the dhencha, and Mesorhizobium loti, they forms this symbiotic relationship with lotus, and also you can see Bradyrhizobium japonicum. So Bradyrhizobium japonicum basically helps in fixing the nitrogen in soybean or glycine max. And so this Rhizobium leguminosarum is responsible for fixing nitrogen in case of peas and lentils. So Bivalve fascioli is responsible for fixing the nitrogen in case of Phaceolus species, which is basically dry bean or string bean, and Bivalve VC is responsible for peas and lentils. So you can see that specific microorganism is responsible for fixing the nitrogen in specific host legume. So specific host legume. So let us move ahead and see what is the symbiotic fixation with non-legumes. Obviously, there are some there are some nodule forming non-legumes. Obviously, there are 220 species of 8 plant families that are known to develop nodules and to accommodate symbiotic nitrogen fixation when their root hairs are invaded by soil actinomycetes, and these soil actinomycetes basically from the genus Frankia. And on a worldwide basis, the total nitrogen fixed in this way may even exceed that fixed by agricultural legumes, because of their nitrogen fixing ability of certain tree actinomycetes association are able to colonize infertile soil and as well as newly formed disturbed lands. So which may have extremely low fertility as well as their condition that limit the plant growth. And remember that certain cyanobacteria are also known to develop the nitrogen fixing symbiotic relationship with green plants, and one involves nodule formation on the stem of Gunnera and angiosperm common in marshy areas of the southern hemisphere, and in this association cyanobacteria of the genus Nostoc can fix 10 to 20 kg of nitrogen per hectare per year. And so let us see some nodule forming non-legumes. Some examples are given, Gelnus alnus, then Mircea, then Casuarina, and all these are nodule forming non-legumes. So they form the nodules for fixing the nitrogen, and also symbiotic fixation with non-legumes. So fixation without legumes. So let us see fixation without legumes. So among these most significant non-legume nitrogen fixation system are those involving the cyanobacteria. Among all the non-legumes, the most important nitrogen fixation system is basically occurs to cyanobacteria, and one system of considerable practical importance is Azolla Anabaena complex. Now, Azolla is a water fern and Anabaena azole is basically a algae, which, and this Azolla and Anabaena complex or symbiotic relationship basically produces this biological nitrogen fixation, and it flourishes in certain rice paddies of tropical and semi-tropical areas, and the Anabaena cyanobacteria inhibit cavities in the leaves of the floating fern as well, and fix quantities of nitrogen which is comparable to those of more efficient Rhizobium legume complexes. So, it is very important, and you can see here the Azolla Anabaena complex. This is an Azolla fern, and obviously at the bottom of the Azolla these Anabaena colonies can be found, and basically this type of symbiotic relationship can be found in paddy soils, and which is also responsible for fixing atmospheric nitrogen in this type of soils. So, non-symbiotic nitrogen fixation, certain free living microorganisms also present in soils and water, and also they are able to fix nitrogen. Because these organisms are not directly associated with higher plants, the transformation is referred to as the non-symbiotic or free living. And there are two types of non-symbiotic nitrogen fixation. One is fixation by heterotrophs and another is fixation by autotrophs. So, several different groups of bacteria, in case of heterotrophs, several groups of bacteria are known for non-symbiotic nitrogen fixation. Examples are Azotobacter, Azospirillum, and then Beijeringia. And in case of anaerobic condition also, the Clostridium bacteria also known to fix the nitrogen. So, these organisms obtain their carbon either from root exudates or in the rhizosphere or by saprophytic decomposition of soil organic matter, and they operate best when the nitrogen is limited. Obviously, when the nitrogen is abundant, why they should fix the atmospheric nitrogen. And fixation by autotrophs, obviously in the presence of light, certain photosynthetic bacteria and cyanobacteria are able to fix carbon dioxide and nitrogen simultaneously, and the contribution of photosynthetic bacteria is obviously uncertain, but that of cyanobacteria is thought to be some significant, especially in wetland in case of rice paddies I have already told you. So, in some cases cyanobacteria contribute a major part of the nitrogen needs by the rice, but non-symbiotic species rarely fix more than 20 to 30 kg of nitrogen per hectare per year. So, here you can see the levels of nitrogen fixation from different systems. Obviously, symbiotic, you can see different symbiotic nitrogen fixation, obviously they varies from 100 to 500 kg of nitrogen per hectare per year, and in case of alfalfa you can see 150 to 250. So, the symbiotic nitrogen fixation is known to produce high amount of nitrogen through symbiotic relationship. In case of non-legumes, obviously some little bit amount, you can see casuarina there is producing 10 to 20 kg of nitrogen, and in case of azotobacter and azospirillum they are producing obviously less amount, that is 5 to 30 kg of nitrogen per hectare. Azolla cyanobacteria relationship, they produce huge amount 150 to 300 kg of nitrogen per hectare, and some in case of non-symbiotic bacteria like Clostridium, they produce they can fix 5 to 20 kg of nitrogen per hectare per year. Now, rhizobacteria are bacteria adapted to the life in the rhizosphere are called rhizobacteria, and rhizobacteria that benefits the plant growth and development are known as the plant growth promoting rhizobacteria, PGPR. These are very important, and many PGPR bacteria belong to the genera of Spirillum, Rhizobium and Azotobacter, and these microorganisms obtain their energy supply for their nitrogen fixation from the exudates of the plant roots, and these microorganisms are nowadays being utilized in different formulation for application into the field for for several beneficial action for the growth of the plant. And this is my couple of slides. So, the factors affecting nitrogen fixation, obviously the natural population of the rhizobium soil is too low to fix nitrogen. So, in these conditions special mixture of the appropriate rhizobial bacteria may be applied into the as an inoculant either by coating the legume seed or by applying the inoculant directly into the soil, and the legume rhizobium association generally function based on soil that are not too acid, and the soil should be well supplied with essential nutrients and have sufficient amount of organic matter, and high amount of nitrogen sometimes in soil inhibits the nitrogen fixation. And remember that one thing, that increasing amount of nitrogen, when you are increasing the nitrogen fertilizer application, obviously the nitrogen fixed by nodulated bacteria getting reduced, although the total nitrogen in the plants may remain fixed. However, the there is a decrease in the nitrogen fixed by nodulated bacteria when we increase the nitrogen fertilizer. So, guys, we have covered the one of the major process of nitrogen cycle, that is biological nitrogen fixation. Obviously, we cannot complete all the important details, the nodulation process, which is very much intricate and requires some complex signaling process. We did not have time to discuss all of these, but I hope that you got a basic idea about the biological nitrogen fixation, and I will encourage you to go ahead and consult several books to get a better knowledge of in-depth knowledge of biological nitrogen fixation, because this is very important for soil and plant growth. So, guys, thank you, and let us meet in our next lecture with when we will start soil phosphorus and potassium. Thank you.